All right, so we want to welcome everyone to this seminar. The general topic has to do with the intersection, the interactions between chemistry and astrobiology. You will hear the most specific topic pretty soon. But as we normally do, I introduce my co-host. Today, my co-host is Jean-Juan Bay. I have a sense, Jean-Juan, that we've done this before. I have a deja vu feeling. Haven't you been a co-host on something with me before? I think so.
I think for Environmental Fridays,
Okay.
for one of them.
Okay. We've done this before. Yes, so very good. So John is a student here at Andrews University, a junior biochemistry major. He graduated from Berrien Springs High School. I think it was now maybe about 2 years ago, you took OChem lab.
Yes.
Very enthusiastic about his research project. And I think right now, are you still doing a project with Dr. Novak?
Yes, I actually have to contact you about collecting water samples in the Benton Harbor area.
Okay, all right. We'll do that. We'll do that. Very cool. So he wishes to pursue medicine. He's a hometown guy, you know, from Berrien Springs, Michigan. He wishes to pursue medicine after undergrad, and he enjoys running and hiking. Are you starting to run already? It's kind of.
Oh yeah, just indoors for now.
Okay. Okay. All right. Sounds good. All right. So, John, you could take it over and introduce our speaker.
Okay. Dr. Tori M. Hoehler graduated from the University of North Carolina with the Bachelor's of Science in Chemistry with the highest honors and highest distinction. Afterwards, Dr. Hoehler obtained his PhD in Marine Chemistry and Biogeochemistry from the University of North Carolina. He began working at NASA in 1998 at the Ames Research Center as a National Research Council postdoctoral fellow, and now he's a research scientist in the exobiology branch. He's also the recipient of the H. Julian Allen Award, one of NASA's Ames' highest honors for his research in microbial life under extreme energy limitation. In addition, Dr. Hoehler is a recipient of NASA's Exceptional Science Achievement Medal and NASA's Outstanding Leadership Medal. Dr. Hoehler's research interest includes microbial biology, ecology, and biogeochemistry, surface and low energy ecosystems, and planetary habitability. Today, Dr. Hoehler will be presenting on energy flow at the biosphere level, observations from Earth and implications for the search of life beyond Earth. This topic is crucial as studying the bioavailable energy flow on Earth allows us to transition from knowing what's required for habitability to detecting if some place is habitable. Lastly, a fun fact about Dr. Hoehler is that he was in a documentary called Aliens of the Deep, directed by James Cameron. Now, please join me in welcoming Dr. Tori M. Hoehler.
Thank you, John. I'm not sure how you dug up all that information, but anyway. Thank you all very much for the invitation. It looks like this has been a really amazing seminar series, and I'm happy to be a part of it. I appreciate that. So let me share my screen so that we can jump in. And I assume that one of you will yell at me if you can't see my slides. Otherwise, we'll just jump in. So as John said, you know, the title of the talk is Energy Flow at the Biosphere Level. Ultimately, this is a talk about how life on Earth relates to energy, but I wanted to start with a little bit of context to help you understand why it is that this work gets done at NASA. And this image in particular is one of my favorite from NASA. So this was taken by the Cassini spacecraft around Saturn, and the small blue dot that you see in the lower right-hand corner, that's us. That's Earth from about 800 or 900 million miles away. And I like this image because I think it puts us in the broader context of the solar system. And in particular, we've learned over about the last 3 decades or so of planetary exploration that Earth is just one of at least half a dozen ocean worlds, meaning worlds that have substantive bodies of liquid water in the present day. In general, in the subsurface, unlike Earth, but nevertheless, we're not the only ocean world in the solar system. And each of these has its interesting features, but I want to focus on this little one in particular. Enceladus is a moon of Saturn. It makes kind of a nice case study, and arguably it's the place where we understand the most about the chemistry of that moon. So a little bit about Enceladus. It's very small, so it's seen here for scale next to the United Kingdom. Its small size means that it has a very low surface gravity, only about 1% that of Earth. And that means that as the moon accreted, that is when material fell in and ultimately formed the moon, the amount of heat generated through that process was relatively small. And similarly, because of its small size, it has a much greater surface area to volume ratio than Earth, which means it cools much more rapidly. Its capacity to heat its interior through radioactive decay is much smaller than that of Earth. And so we expect that its internal heat, this combination of accreted heat and radiogenic heat, is going to be very small. Enceladus is also far away. It's about 10 times as far away from the sun as Earth is, which means it receives about 1% as much solar radiance as Earth does. And the combination of those two things leads to the expectation that this should be frozen solid. There's really not enough light to keep it very warm at the surface, and there's not enough heat from inside to keep it warm inside. And the expectation is completely that this should just be an ice ball. And as the Cassini spacecraft that took that beautiful image from Saturn earlier got closer, here's what it revealed. So if you look at the northern hemisphere, the top of the picture of Enceladus, what you see is a cratered surface that looks kind of like our own moon. And cratering is essentially an indication of a geologically inactive surface. There's no process that refreshes the surface and removes those craters. And so the indication is that northern surface of Enceladus is a little bit on the old side. But contrast to that, what you see in the southern hemisphere, almost no craters are visible. And the implication is that this surface is replaced and replenished on a relatively rapid timescale, maybe millions to 10 millions of years or so. And as the Cassini spacecraft got closer, here's what it saw. So if you look at the little globe on the left-hand side, this is an expectation of what the surface temperature of Enceladus should be, simply based on solar radiance. So just like our own Earth, you expect it to be warmer near the equator, colder at the poles, and look at the temperature scale there. So what you really expect at the surface of Enceladus is ice that's frozen to about 80 Kelvin and even colder than that at the poles. Cassini carried an infrared spectrometer that essentially was capable of mapping the heat flow across the planet's surface, or the temperature across the planet's surface. And instead, what you see is that it's very bright in the southern hemisphere, meaning the temperature is significantly higher than expected. And it zoomed in even closer to the little object that you see on the right. That's essentially a crack or a fissure in the surface, and the peak temperatures there are more than 100 degrees warmer than would be expected from simple solar radiance. And so the implication is this is a geologically active world. There's much more heat coming out than would be expected, and it's expressing itself at the surface. And perhaps most interestingly, as Cassini turned its cameras towards Enceladus, what it observed in this backlit image is actual plumes of gas and ice emerging from the subsurface of Enceladus, and many people believe emerging from a subsurface ocean. Cassini carried a couple of instruments that were capable of sampling this. Now, it wasn't known at the time that Enceladus had this kind of activity, so nothing about the instrument suite was designed to make any measurements there. But fortuitously, there was a mass spectrometer on board, very simple one, mass range of about 100 with unit mass resolution, but sufficient to see that material contains abundant water, CO2, hydrogen, methane, ammonia, and some organics of maybe C4 and greater. We can't tell very much more because of the limited mass range. Perhaps also some hydrogen sulfide. We haven't observed any phosphorus, and that's simply because the sensitivity of the instrument package to phosphorus wouldn't have been enough to detect it even in our own oceans. But I present all of this because in these group of volatiles, many of the ingredients required for life are present. We don't know about phosphorus, but we do know about most of these other items. There was another instrument called the Cosmic Dust Analyzer, essentially analyzes particles about the size of smoke, so in the micron range or so, and gives some compositional information. It was able to tell that in this material that's emitted, there's sodium chloride, just like in our own Earth's ocean, and enough so that an inference can be made that there's a subsurface ocean that's about 10 to 40% as salty as Earth's ocean is. And that's significant for reasons that I'll get into in just a minute. The CDA also observed some silica and nanoparticles, and these are interpreted as potentially being evidence of hydrothermalism, so hot water reacting with rock and generating silica that then precipitates out into small nanoparticles as it hits a colder and more alkaline ocean. And so all of these things together have painted this really interesting picture. And there's a colleague of mine, a planetary scientist, who likes to say that Enceladus is far more interesting than it has any right to be. So the interpretation of all of these data is that Enceladus hosts a global ocean of perhaps tens of kilometers thick beneath an ice shell that is about 10 to 20 kilometers thick. The salinity that we see in the ocean, so here on Earth, the salinity of our ocean basically comes from dissolution of continental rocks by acid volatiles that come from volcanoes. Something similar is going on in Enceladus, and you can infer that there's been pretty extensive interaction of water with rocks, consistent with a style of water rock reaction called serpentinization. That's important because although the fluids contain volatiles that are necessary for life, so do the rocks. And in many ways, the rocks also contain energy that are important for life. And so the idea that there's been this water rock reaction is conducive to the notion that life may be supplied with resources. There's evidence of possible hydrothermalism. I'll come back to that. And remarkable, I think, for a spacecraft that never was designed to make such measurements. We actually have some constraints on pH. It looks like the ocean is moderately alkaline, on dissolved carbon, nitrogen, and sulfur, and even on bioavailable energy as the combination of hydrogen and CO2. So on Earth, those two things can be metabolized to form methane by a kind of bacteria called methanogens. So in parallel with all of what was being learned about Enceladus, and each one of these observations is positive with respect to the potential for life, we were making similar discoveries on the other ocean worlds. So much so that a few years ago, 5 or 6 years ago, got this directive from Congress. The committee directs NASA to create an ocean worlds exploration program whose primary goal is to discover extant life on the world. So this represented something of a phase transition because a lot of the observations made thus far had been interpreted in the sense of, could life be possible there? Have we learned anything that dissuades us from thinking that life might not be possible? Now with this directive, we have to kind of shift our thinking and instead begin to ask, how would we actually go about seeking evidence of life? So not just could life exist, but if it is there, how robustly and abundantly inhabited are these worlds? What evidence should we seek of life, and what kind of environmental context should we establish in order to interpret that evidence? And in specific, this means what measurements do we make and with what kind of performance requirements? Do we fly a mass spec? Do we fly a microscope? Or something entirely different? And in any of those cases, what kind of requirements do we levy on those things? And importantly, and really the subject of this talk is, can or should we inform these choices by reference to life on Earth? So after all, this is the only example we have of life and the only place in which we can understand how environmental context bears on life and its observable residuals, meaning the signs of life that we might seek. When we do this, I think we have to be very careful that we also acknowledge how different these places are and understand the implications of those differences for life and its potential expression on these other worlds. So let me jump into looking at life on Earth and how it deals with energy. And I think the first and most significant lesson that we can learn is just by looking at the world around us. So the image that you see there in the lower right, I actually find this kind of mesmerizing to look at. This is a 5-year history of remote sensing measurements of what is essentially chlorophyll on the land surface and in the ocean. So we're watching the coming and the going of photosynthetic biomass. And the first takeaway is a spatial one. That is, if we look around us, life isn't just either present or not present. It varies in a continuum of orders of magnitude. So on land, the abundance of photosynthetic biomass varies from roughly 0 in the hyperarid core of the Atacama Desert to better than 50 kilograms per meter squared of biomass in the dense jungles of the tropics. And in the oceans, if you look at the color coming and going, the purple colors are about 0.02 milligrams of chlorophyll per meter cubed. So that's a measure of how much phytoplankton biomass is there, to about 20. So that's 1,000-fold variation in the abundance of photosynthetic life. And on land, that variation is driven primarily by water availability. In the oceans, it's driven primarily by the availability of nutrients like nitrogen, phosphorus, and iron. And so I think it's this kind of quantitative perspective that we should carry with us in understanding how to look for life on other worlds, that dramatic variations can be driven by environmental context. The other thing that I want you to notice, though, is that every time you see that little yellow scale bar go around in the picture of the globe, that's a year. And so really what we're seeing is the Earth and its biosphere essentially breathing. We're seeing the standing stocks of life at the surface of the planet, made and unmade, on year-long timescales. And on land, the average turnover time for all of the plant biomass on land is about 7 to 8 years. In the oceans, it's about 3 to 4 days. The entire standing stock of phytoplankton in the ocean is replaced about every 3 to 4 days. And this is what provides the context for how we think about what an inhabited world looks like, robustly, abundantly inhabited. And the reason for that is that we live in a world that's absolutely replete with resources, and in particular, replete with sunlight that provides life with abundant energy. But if we go beneath the water, all of a sudden we see a different picture. So what you see plotted now on the y-axis is depth in log of meters beneath the seafloor. And on the x-axis, it's log of cell abundance. So there is 8 orders of magnitude in the abundance of cells represented along the x-axis. And what you see is that as you go down with depth and as you go to more and more nutrient-poor portions of the ocean, there's a more than 10 million-fold variation in the abundance of cells that are supported in these different places. And that variation is driven not by water, which is there all the time, and not by nutrients, which are plentiful, but by the availability of energy. And this, I think, is where we need to begin looking in order to understand the potential for how robustly life can be expressed on these other ocean worlds, because energy is very likely to be limiting. So this hasn't been lost on the planetary science community, and there have been efforts to make estimates of biomass abundance on, in this case, Enceladus, based on the amount of energy. And these estimates, broadly speaking, have partitioned energy in 2 different places, the 2 sort of productive things that life can do with energy from the standpoint of leaving evidence of itself. One is biosynthesis, the production of new material that ultimately enters the dissolved phase. The other is the support of standing biomass. And that's what I want to draw your attention to. It's the column on the right. So what you see represented in the red numbers there are the span of uncertainty. That's about 9 orders of magnitude of present uncertainty in how much life this body could support. And if you're trying to design an instrument package to go and look for life on this world, understanding how much life is there is absolutely critical to assessing the performance requirements of those instruments. The range that's represented here goes from perfectly detectable by a spacecraft to absolutely no chance of ever seeing that. And so the idea is we really need to try to understand the interaction of energy and life a little bit better in order to try to resolve this more finely. So when you do a calculation like this, implicit in doing so is the idea, first of all, that you understand how much energy there is, and both of these studies focus primarily on that, but also how life uses that energy. How do we equate energy to standing biomass? And of the 9 orders of magnitude uncertainty that you see, I'd say about half is attributable to uncertainty in the energy availability, and the other half is attributable to uncertainty in how to deal with, how to equate life and biomass to energy. So that's really what I want to focus on. How do we make that transition and that calculation? And I want to do 2 things in the remainder of this talk. The first is I want to try to assess in quantitative terms how much less energy is available to a biosphere that lacks access to light energy. So I don't think it will surprise any of you if I say that our biosphere gets a lot more energy because we have access to sunlight. This is the only place in the solar system where sunlight shines directly into a liquid water environment. and that matters. But I think it may be surprising how much that matters. And so I want to try to quantify that for you. The second thing I want to do is ask, what's the relationship between energy flux, that is how much energy through time, and the biomass that it's capable of supporting? And I'll try to do this at the level of individual organisms, but also at the level of the biosphere. So let me try to give you an example. And here's an energy budget constructed for the marine biosphere. We're able to do this because inherently energy flow on Earth and life is inherently tied with carbon cycling, either as the capture and fixation of CO2 by photosynthesis, or the respiration of that CO2 as it's passed into the heterotrophic part of the biosphere. And carbon cycling is so important to understanding global change that there is a robust body of research that's tried to quantify these flows. And so the available energy for photosynthesis that strikes the surface of the Earth in the wavelengths that can be accessed by photosynthetic biology is about 40,000 terawatts. So really only about 2.5% of that is actually used by our ocean photosynthetic community, that's phytoplankton. So we are absolutely not energy limited on Earth. There is more than enough energy. The phytoplankton use that, they grow rapidly, they die, and that organic matter is passed into the water column, and then it becomes available for organisms like us, organisms that respire organic matter with oxygen. That's worth about 60 terawatts of power to the ocean water column. Most of that productivity, that organic matter, gets consumed in the water column. A small fraction of it passes down and ultimately is deposited at the bottom in the form of sediments. So in the upper meter of that sediment, that energy flux, that flux of organic matter, is worth about 3 terawatts of power. And in the sediments beneath that, only a very small fraction survives. And what's left is about 0.01 terawatts of power. So what you see is the diminishment of energy as you move through this ecosystem over many orders of magnitude. That's what happens in a photosynthetic world like ours. But what about the possibility of hydrothermal vents, of the planet itself serving as an energy source? And when many people think about these ocean worlds and the potential for life there, the picture that they have in mind is of hydrothermal vents. So for those of you who don't know what these are places, you can think of them as equivalent to underwater volcanoes, except what's happening here is that water is circulating down through the crust. It's becoming superheated at hot spots in the crust. And as it does that, it reacts and equilibrates with rocks, which have a very reducing character, and it emerges at very high temperature. And when it does, it carries a variety of reductants into an oxygenated ocean. And that essentially represents a flux of energy. That's energy waiting to be used by largely bacteria and then higher organisms. So it turns out there's enough written on this that you can construct a budget for this as well. The major reductants that come out of these systems are hydrogen sulfide, methane, hydrogen, and ferrous iron. And if you add all of those up, if you consider all the energy available in respiring each of those things aerobically, the power available in that system is about 0.03 terawatts. And so if you compare that to what we saw on the previous slide, that's kind of comparable to that very last little residual of energy, the 0.01% or so of energy that makes it from the surface down to those deep sediments where everything's already been chewed up. So to put this in context for you, on the y-axis here, you see power. So that's units of energy through time on a log scale. So from about 100 watts to 10 to the 16 watts at the top of the scale. So if you do the math, you have a power throughput based on your diet of about 100 watts, which makes you roughly equivalent to an incandescent bulb. Global photosynthetic energy capture on Earth, as I just showed you in that budget, is around 10 to the 15 watts or so. And the energy that's available through these hydrothermal venting processes in aerobic terms sits sort of in the middle of that, about 2/3 of the way from you to global photosynthetic energy capture. And if oxygen isn't in the picture, because after all, the oxygen in our oceans comes from photosynthesis. So if you took that out of the picture, the energy available in hydrothermal processes would drop by about an order of magnitude or more, because the energy now in oxidizing those things is considerably less. So this is significant, right? The amount of energy that's available in a geochemistry-only world is roughly halfway between the energy that you as an individual consume and the energy that our photosynthetic biosphere as on the whole consumes. And that's got to have significant impacts on the potential for life or the abundance of life on these worlds. So I want to express these same data on a slightly different scale. Now I'm going to do it on a volume normalized scale. So basically what I'm doing is calculating the amount of energy flowing through a system divided by the volume of that system. And the reason for doing this is a fewfold. One is that the oceans on these ocean worlds are much, much deeper than our own ocean. So our own ocean is on average about 4 kilometers deep. On these ocean worlds, the oceans may range from 20, 30 to maybe even 100 kilometers deep. So you're diluting an energy flux into a much larger body of water, and we want to be able to account for that. But also when we put it in volume normalized units, we're beginning to come closer to something that looks like a concentration. In this case, watts per liter, but if we begin to think of how many cells that may support or how much biomass is generated, now we can also think of that as like a concentration, cells per liter or moles per liter. And that's the kind of information that we need to be able to design instrument performance requirements. So we can plot the energy flow on this scale through our sunlit zone of our own ocean. That's the ocean photic zone, also through the deep ocean. And if I plot the geochemical only Earth, that is an Earth that has no photosynthesis, but fueled entirely by this reduced energy flux from hydrothermal vents, it's many orders of magnitude below anything that we can access in our surface ocean or even in our deep ocean. And the implication is that neither of those two places is particularly good as a representation of what we might see as evidence for life on one of these other ocean worlds. And in fact, the estimates that have been made of energy flow on Enceladus and Europa fall in this range. And so our geochemical Earth, our geochemical only Earth, sort of represents the upper end of that spectrum, and it only gets worse from there. So what I want to ask about is, what is the implication of this for abundance of life in these systems, if it exists there? How do we equate energy availability to life? And this goes to the idea of mass-specific power. So mass-specific power sounds like it's sort of a complex concept, but it's really not. The idea is simply that life requires energy simply to maintain a metabolic status quo. Not new growth or anything like that, but just to keep the status quo. And as an example, I think that I calculated that since I was 18, I've consumed something like 100 gigajoules of energy. And yet I've been approximately the same version of me since then. No net growth, about the same. I've required all of that energy just to support a metabolic steady state. And you can apply that same thinking to any kind of life on Earth and ask, how much energy does that organism require? And how much does it weigh? And if you divide one by the other, you get a quantity called mass-specific power. And the idea is that mass-specific power is a way to equate energy to biomass. So what you see represented here, in order to try to understand how this plays out on Earth, we looked first to a large body of research that's been done on individual organisms. So these kinds of measurements, what's the metabolic rate of this organism? How much does it weigh? What's the mass-specific power? Have been the basis for a very large amount of study. And you see a compilation of as many studies as we could find. There's something like 10,000 individual measurements represented on this plot. There's a large abundance of them in the area sort of between 10 to the minus 4 and 10 to the 2 grams of body mass. That's the x-axis. And that's because much of the focus has been on animals and animal physiology, organisms that are of interest, organisms that are accessible and amenable to study. That's where the bulk of the study has been. If we try to ask about life on these other worlds, we're actually much more interested in what is likely a microbe-only world. And so how do we extrapolate the information that we see in a plot like this to a microbe-only world? So let me try to walk you through this a little bit. What you see represented here is something like 22 orders of magnitude in body mass from the smallest bacteria up to the largest whales. And the mass-specific power of each of those individuals is plotted on the x-axis, again, on a log scale. All of the trend lines that you see, so the colored lines represent a very large body of research that's been done on an apparent mass dependence in this quantity of mass-specific power. So meaning that as an organism gets larger, its mass-specific power, its metabolic rate per unit of mass goes down. And this is well documented and well explained at the level of individual groups of organisms. So certain families of birds or mammals or things like that. But if you look across the whole plot, there really is no mass dependence evident. And so we have this sort of conundrum that there's clear mass dependence in these individual taxa, but not at all in life overall. The other thing that I think is really interesting to note here is that there's about 7 orders of magnitude represented on the vertical scale in mass specific power. And life overall occupies most of those 7 orders of magnitude. So that means some organisms are capable of operating with 10 million times smaller energy throughput than some other organisms are or are capable of. And that is also interesting because if you think about it, if you take any one of those trend lines that's represented there and try to extend it across the entire mass range, you'd very quickly go beyond the boundaries of this plot. And the boundaries of the plot are chosen for a reason. They represent to me, roughly speaking, physiological or biochemical level limitations on what might be possible. So for example, what's the fastest that enzymes can work? How densely can you pack an organism with enzymes, and what therefore is the fastest rate of metabolic throughput that you can support? Similarly, at the bottom end of the spectrum, what's the lowest rate at which you can repair or fix or replace your essential biomolecules against the natural attrition that those things suffer? And those two things establish biochemical boundaries that transcend the specifics of what organisms look like. They take effect at a biochemical level. And that, I think, is what we see playing out here, that life is held within these sort of 7 order of magnitude bounds on mass specific power. And that as we move along the mass spectrum, we see life constantly reorganizing its more superficial aspects in order to stay within those bounds. So this doesn't really help us very much with our uncertainty. If you think about it, this is 6 or 7 orders of magnitude uncertainty in how to equate energy to biomass. So what is true in this plot is, despite those 7 orders of magnitude, the large majority of the data cluster in about 5-fold of a mean value, represented by that black line and the gray shaded region that sits almost intermediate between those bounds. And so the question is, does that give us anything? Does that median value give us anything to work with? So briefly summarizing, here's what I think we can take away with this. A few observations, a few caveats. The first is that across all organisms in that data set, the mass specific power spans about 6 orders of magnitude range, but most of it lies very close to a median that sits in the middle. Mass dependence is evident within specific taxa. Those were those colored trend lines that you saw, but not in the data set overall. Excuse me. And I think what this means is that it's subject to upper and lower bounds that really are a result of biochemical limitations and therefore are applicable across that full range. So what's important to realize about this data set is it's not at all a representative sampling of Earth's biodiversity. It's representative of what we could find in the literature. And there's a real overrepresentation of animal species in here, and in particular mammal and bird species and fish, relative to what Earth's biodiversity really looks like. So it's a biased sample set. It's also not at all weighted relative to the biomass distribution on Earth. So statistically in that plot, most of the values that you see are generated by animals, and the biomass on Earth lies mostly in plants and in microorganisms. And so what you see statistically in the plot is not a representative sampling. And these experiments are all conducted under very specific conditions, so very well-defined conditions and very limited durations. And so they don't encompass any ecological effects. They don't encompass life cycles, birth, growth, death, anything like that. And so the question becomes, how useful are they in actually making any predictions about what happens at the biosphere level? So because of that, I sought to try to make an assessment of mass-specific power as it's expressed in the environment in the major components of Earth's biosphere. So what you see represented here now is the same plot that I showed you earlier, where we talked about energy flux through these different reservoirs of the biosphere. But now represented on this plot are not only energy flux, that's the red numbers that you see, the 1,057 and so forth, but an estimate of the biomass. So there was a publication a few years ago that tried to estimate the total biomass in phytoplankton, in all of the life that sits in the ocean water column, including fish and bacteria and so forth, in sediment life in the upper meter, and in deep sediment life. And finally, in green is the quotient of those two. So it's the power divided by the mass. So this is the green number, mass specific power. Watch what happens as you go from phytoplankton, 770 milliwatts per gram carbon, to life that exists in the water column, 11, to life that exists in the upper meter of sediment, 0.65, and to life exists at the very bottom in the deep biosphere, 0.0028. There's about 4 or 5 orders of magnitude decline from the surface of the ocean to the deep sediments in mass specific power. So whereas at the organism level, we see about 6 orders of magnitude range that's physiologically possible, what we see expressed in the environment is that most of that range is actually taken up and expressed by one or another class of organisms. And I want to focus a little bit on this particular group right here, the sediment column. This is where I did my dissertation research. This is what I understand sort of the best. And so in the plot, what you see represented here, the y-axis is depth beneath the seabed. So this is depth in the sediment column in centimeters. The x-axis is 2 things. SRR is sulfate reduction rate. So that's a particular kind of microbial process. And these rates are expressed in cell-specific units, femtomoles per cell per day, and that's a log scale. And with those rates, we can actually equate them to mass-specific power by making an estimate of cell mass and of how much energy is represented in these rates of cell-specific sulfate reduction. And so, of course, that's also a log scale. And that log scale spans about 4 orders of magnitude. So what you see here as you move from the top of the sediment column at 0 down to about 3 meters depth is that the mass specific power decreases by about 3 orders of magnitude from the top of the sediment column to the bottom of the sediment column in relatively continuous fashion. And this particular part, if you look below 1 meter, that's what the deep sediment community has operationally defined as the deep biosphere. It takes about 2,500 years of sedimentation for sediments to get down to this depth. And so significant time has passed, and the decline from a relatively high value of mass specific power down to this very basal level occurs over that 2,500 years or so, and then remains relatively constant, even as the sediment continues to age, even as the amount of organic material gets poorer. And so I think what we're seeing here is a natural expression of what appears to be a physiological lower limit. Now, if I take that deep biosphere component and I use a methodology for trying to understand how rapidly those cells are turning over, and turning over means they're consumed in one way or another and then replaced by new growth. What you see represented in the plot here now is the log of the cell-specific carbon turnover time, and on the right-hand side, an estimate of turnover time in days, weeks, months, and so forth. The red plot, the red points are typical cultures. So this is what we're used to seeing in the kind of physiology experiments that teach us how to understand energy metabolism in life. The green points are surface sediments, and the cluster of points down here, circled in red, this again is the deep biosphere. And what I want to draw your attention to is the implied turnover times for these cells are on the order of hundreds to thousands of years. So here's the implication. We can do this set of calculations for mass specific power for a whole bunch of different portions of the Earth's biosphere. So let's start at the right-hand side of the plot. The black point that's circled in red, that's this calculation done for our biosphere overall. The gray shaded region with the line through it, that's the same median value from the organism level plot of mass specific power carried into this plot. And so actually that median value predicts the mass specific power and in fact the mass of the biosphere quite well, to within a factor of a few, which I think is kind of remarkable. Most of the other things that we can measure, so land plants, for example, soil biota, humanity, all sit within that sort of 5-fold range of that median. And the exception comes when we get to the marine biosphere. So these are the different portions that I showed you of the marine biosphere. The blue point at the top, that's the phytoplankton. The yellow point is the ocean water column. The orange point is the upper meter of sediments. And the green point is the lowermost sediments of the deep biosphere. And what's driving this, whereas almost every other component of the biosphere that we look at sits close to this mean value, the ocean does not. And what's driving this really is a turnover time. So the phytoplankton and the upper portion of the water column are such tasty food for creatures that would graze on them, that entire community globally is turned over on the order of 3 to 4 days. And at the bottom end of the spectrum in the deep biosphere, that cellular population goes hundreds to thousands of years on average before turning over. And so it's remarkable to me that not only does the biosphere lie close to the mean that's expressed at the organism level, but it also expresses much of the possible range, about 4 to 5 orders of magnitude of possibility as established by organism physiology. So let me try to bring this back around to this idea of volumetric energy flux. So here again is log watts per liter on the x-axis for reference, our ocean photic zone and the deep ocean. And then in blue, the range of estimates that have been made for energy flux on Enceladus and Jupiter's moon Europa. And the implication here is that there's such a large gap, about 3+ orders of magnitude between what we can see in our own oceans and what's envisioned for these places, that our oceans really are not a very good analog, not a very good place for understanding how life might work in such an energy poor environment. But we do have something that we can point to, and that is the work that's been done in deep sediments, in part some of the work that I just showed you, and in parts efforts to go out and drill hundreds of meters down into these sediment columns to access life that is truly energy starved. So that begins to overlap with the upper end of the range of estimates for Enceladus and Europa, and it becomes our best analog for understanding what life might look like in these places. So to try to pull all of this back together, what you see here is a plot with the same x-axis that I just showed you, log of watts per liter. Now I'm going to try to equate this to biomass using what we've learned about mass-specific power on Earth. So the y-axis is log of cells per milliliter. And the larger that number is, the easier it would be to detect that sort of life. So a couple of points of reference. In Earth's deep oceans, the cells per milliliter are about 10 to the 5 to 10 to the 6 or so. So representing sort of the upper third of that plot. What's been proposed for space flight is instruments that have a capability to detect about 10 to the 2 cells per mil, so roughly about halfway along this plot. Now, let me try to walk you through this. All of the points that you see are measurements made in sediments. They're measurements that pair an estimate of the energy production in those sediments with the amount of cells that are supported per liter of sediment. And there are 2 different environments. The blue points are the one that I just showed you a few minutes ago from a place in Denmark called Aarhus Bay. The orange points are much deeper cores taken from the Peru margin. So these go down to almost 100 meters or more. And I've plotted them here for cell abundance versus energy availability in log of watts per liter. And what you see is a trajectory. So as we move from right to left on the plot, meaning that we're decreasing the amount of energy that flows through the system, at the same time, the number of cells that are supported in those environments decreases. But they do so in an interesting way. So if you look at the dashed lines on the plot, what I've done is to calculate the amount of energy required to support a standing population of cells that turns over, in the case of the upper line, with a time scale of 1,000 years, and in the case of the lower line, with a time scale of 1 year. And so what you see is that initially, so if you look at the blue point on the far right, that's the uppermost portion of the sediment column in Aarhus Bay. So this is only about 10 centimeters deep. That point plots with a turnover time of about 1 year or so. And as you move down in depth in the sediment column, that means you're moving left to right on this plot, you see that the number of cells stays approximately constant even as the energy flux goes down. And the implication is that the mass-specific power is going down to accommodate that until it hits this upper trend line, the upper dashed line that represents turnover of about 1,000 years. And then it gets pegged to that line. And the implication is that's as low as life can go. And now any diminishment in energy flux has to be accommodated by a diminishment in cell biomass. So the orange points in particular begin to impinge on the upper end of the range that we estimate for Europa and Enceladus, and actually give us cell numbers that are not bad, things that we could detect with a microscope in spaceflight, things that are really not very far off what we see in Earth's deep ocean. But that's taking the very, very lowest possible mass-specific power that it seems to be expressed in environments on Earth. In fact, that seems to be physiologically possible in environments on Earth. And I think it remains an open question whether it's possible to have an entire biosphere that lives at that level of mass specific power, or if instead, as suggested in these points, some portion of the biosphere ultimately has to live at a higher mass specific power in order to support that lower portion. And remember, when you look at these points that are clustered around a turnover time of 10 to the 3 years, what's supporting them is organic matter produced by phytoplankton that turn over with a time scale of 3 or 4 days. And I think it remains to be determined where in this plot we could expect the bigger part of a biosphere to lie. So I think I'm probably close to out of time, so let me briefly try to summarize what I've gone through. The first point is that by analogy to a non-photosynthetic Earth, if we stripped away the potential for photosynthesis and just left hydrothermal venting and Earth's own geochemistry as the supply of energy, the energy available to life that may exist on any other ocean world is probably 5 or 6 orders of magnitude less than it is on Earth. That has to have consequences on how much life is there and how robustly evidence of life may be expressed. The second idea is that the concept of mass-specific power, as I've tried to describe it, gives us a way of potentially equating energy to biomass, of looking at these planets, making some estimate of how much energy is available, and using that information to try to parameterize a search for life. And I think there are a few bits of information that I've tried to convey. The first is, at the organism level, where we're looking primarily at an expression of physiology, mass-specific power spans at least 6 orders of magnitude. Life utilizes almost all of the range that appears to be physiologically available to it. At the environment level, global mass-specific power, the mass-specific power of our entire biosphere overall, is actually quite well approximated by the mean that we get out of the organism-level data. Perhaps fortuitously so, given that our biosphere is mostly plants and microbes, and the mean in that plot is driven mostly by animals. Still remarkable that global biosphere is well approximated by the organism level mean. But the components of that biosphere, and especially our ocean biosphere, express a 5 order of magnitude range in mass specific power. And the principal control that I've tried to show you on that range is the rate of biomass turnover, the ecology that is expressed in the environment and is not expressed in the organism level data set. And finally, that the very lowest mass specific power we observe in the environment is in deep sediments. Sediments where biomass turnover times are equivalent to thousands of years. Life is very, very different in these places, but it's these environments and the limited access we've begun to have to them that may provide some of our most important analogs for life on ocean worlds. So with that, let me end. I hope that I haven't taken too much time. And I think maybe we have some time for questions.
Yes, I think we do. Jean-Juan, you want to start?
Oh, okay. My question was, how do you tackle such a unique question? So I know you talked about looking at the mean of the, when you were looking at the humans, archaea, and bacteria and such, and you looked at the mean and you were talking about how it wasn't representative of the, the other factors that could be affecting it. Right. And you switched on to how do you, like, it seems such like a difficult question to answer. What's your thought process to answer such a question?
Yeah, good question. It is a challenging question, and I think I often encounter this in being somebody who studies biology but works for NASA. So I think in the world in biology, we really are in the middle of a revolution in biology right now and the tools of biology. And the revolution is driven by tools, both analytical and computational, that allow us to be ever more specific about how life works at a biochemical and genetic level. And instead, many of the problems like the one that I'm describing here require that we take a more general view on how life works. And so for me, the way to tackle that big plot, all of the genetic diversity that's represented in there, the physiological diversity, the 22 orders of magnitude variation in biomass, is to ask, okay, in the face of all of that variability and difference, what's the same? And what's the same is biochemistry, right? There's this idea of the unity of biochemistry that at the core of things, I mean, we're you and I look different than a slime mold or a microorganism or something like that. But at a biochemical level, we're actually pretty comparable. And so my approach to that plot, almost everything that's been done in the studies represented in that plot is quite specific. Why are the numbers why they are? Can we explain why things cluster very close to a certain mean? Why is there a mass dependence? My question is a different one. How large can the range possibly be? What fraction of the range is occupied? And in particular, what sets the range? And my interest in trying to find these biochemical answers to that question is, that's a handle that is applicable across that entire range. So, for example, the upper limit that I described and maybe went through quite quickly is, a fundamental biochemical limit. If you get to the very, very small size scale where transport is not an issue, diffusion works very rapidly to move materials. Essentially, the limitation on the rate of throughput is how fast are your enzymes and how full can you pack your cell with them, right? And it turns out that if you go and find so studies have been done of the median catalytic properties of enzymes. So if you take that as representing how fast a typical enzyme can be, and you allocate about 1% of the mass of the cell to those enzymes, which is the kind of thing that microbes are able to do, you get a number that's about 100 watts per gram or so. And the fastest growing microorganisms that we have, that we've observed on Earth, so microbes that double on a time scale of about 18 to 20 minutes or so, turns out they have a mass specific power on the order of about 45 watts per gram carbon. And so, to me, remarkable how close life seems to come to what I calculated as sort of a back of the envelope upper limit on what should be possible. Similarly so at the bottom end of the range. And so, I hope that kind of answers the question. I think for me, the toeholds in a difficult question like this are figure out what's the same and see what traction that gets you in trying to address the problem.
Okay. I have a question. It's in a different form. So Earth has life because, how would you complete that sentence? Based on, yeah. Or you could take the opposite approach of whatever planet. Earth has life because, based on what you're doing, your work here, what would you say?
Yeah, so the first thing to say is I'm not an origin of life person. So think of this as speculation on my part. And even for the people who are origin of life people, the question is far from settled. So the generic way in which I would answer your question is, Earth has life on it because a particular environmental context was established that was permissive to life. It is there because it can be, because the conditions, the environmental conditions were right, okay? That's a generic answer. What are the right environmental conditions? I don't know, and I don't think that anybody knows. And what I can say is there are two different camps, sort of prevailing wisdom about how this might work. One camp is very much centered around the idea of ocean vents of the kind that I described. And in particular, a specific type of ocean vent that's relatively rare in our oceans, but should be relatively common in these places like Enceladus. So a style of water rock reaction that tends to generate very reducing fluids, moderately alkaline fluids. And the idea is that a lot of the chemistry associated with that might be conducive to the origin of life. There's a different camp that says you actually need multiple phases. You need mineral phases, you need fluid phase, and you actually need an airborne, a gaseous phase in order to get the diversity of environmental types that you need to do all the chemistry that has to coalesce into life. So if you think about it, there are condensation reactions that have to happen in order to polymerize the sorts of molecules that we use to store information and catalyze reactions. You need to encapsulate those things in membranes. You probably need to start some of your chemistry on catalytic surfaces, like mineral surfaces. And the idea is that if all of those things are required, in particular for the condensation reactions, that an environment that's purely liquid or just liquid and mineral phases maybe doesn't give you the right conditions because it's not very conducive to condensation reactions. So I don't know if that's a good enough answer, but it's the only answer I have for you.
Yeah, it's okay. The meaning like it's an answer. We know it's speculative type thing, but I was also wondering in terms of the framework that you provided us in your talk, if there was some sort of mass power thing that you could have referred
I
to.
understand. I think I understand. So as I think of it, the real relationship that we might be talking about is energy and information, right?
Okay.
So ultimately, we are about storing and replicating information. That's what we do. Everything else about our biochemistry is to support that. And you don't create new information or you don't replicate information without energy to do it. And in fact, you don't even sustain information without energy to do it. Very quickly, entropy will get to work and reduce us to simpler things. And so in my mind, the equation to be made is between information and energy. And so the question to get to your point is there any relationship then between mass and information? So does a certain amount of mass store a certain amount of information, not only in the actual information encoding molecules, which are a very small fraction of our mass, but in all of the proteins and all of the other cellular forms that are necessary to support them? Is there some ratio to be had there? Something that we can understand? That I don't know. I suspect there is. I suspect that it's not only about the mass of the information containing molecules, but some kind of infrastructure needed to support them. How much variability there is in that ratio, I don't know. But certainly there was for any of you who may be interested, there was a study done 10, maybe more than 10 years ago by a National Academies panel that many people refer to as the Weird Life Study. So I think its actual title was the Limits of Organic Life in Planetary Systems or something like that. It was a group of people basically put to work by NASA to imagine life as broadly as they could and say what constraints still exist.
Okay.
They have a list somewhere in that of here are, if we think as broadly as we can, here are the things that we think are characteristic to life and required by life. And they listed about 4 things. So one was a liquid habitat. One was information processing, things like that. The only thing that they listed as an absolute, so they went to pains to say everything that we're listing, if you think hard enough about it, could be off the table. The only thing that's an absolute is energy. And for the reason that I just explained, right? You cannot sustain complexity and information in the absence of energy flow. And that really is the fundamental reason that I come at things the way that I do.
Okay. So while I may not understand everything that you just laid out for us, that you would say is like an underlying principle. What you just said about energy as the most fundamental requirement for life.
To me, it certainly is. And it goes to the question that Zhongwan actually asked just a minute ago, right? How do you attack a problem like this? What if you allow for the possibility that life is different than it is on Earth? You begin by trying to figure out what's the same. And to me, that's something that has to remain the same, no matter what.
Mm-hmm. I see John. John, you have a question in the chat. You could unmute and ask a question.
Oh, sure. I was just wondering about, sorry, I'm not the best with names and catching all the names, but you had mentioned that so the Saturn's moon, right?
Mm-hmm.
We didn't expect it to have all the properties that it did with like
Right.
water and maybe possibility for life and all of this.
Right.
And then that the probe wasn't really equipped. Like you talked about how the mass spec, that was on it then. So like knowing like all of that and then like combined with your research now that you're doing on all of these, looking at so like how would you equip the probe now if you had the chance? And like also how long does it take for like the probe to make a trip? Like if you're going to send one out today?
Yeah, great question. And actually I started out, when I first started making this talk, I was going to put what you just asked as a component of the talk to give you guys a sense of what this is like. So here's a way to think about it. first of all, the time constraint. So there has been a study recently to what you would actually put on a probe to go to Enceladus and look for evidence of life. I think from that study, the probe would launch in the mid-2030s, and it would get to Enceladus in about 2045 and take some time to do its orbital dynamics to get into position. So we're talking about from now, 25 plus years before the measurements begin to be made, okay? And anything that's going to carry instrumentation, it takes long enough to build the instrumentation, to validate it, to system integrate it, and make sure that all of that is done well enough in advance of the launch that you have to add a few years for that. So a measurement that's being made at Saturn is very likely based on technology that's sort of 20, 30 years older than you might like it to be, or than what you have in front of you right now. So with that in mind, the other constraint is you're very far from the sun. So solar panels don't do a lot for you. You get relatively limited energy. You essentially have to carry radioactive batteries with you to generate the power that you need. So the whole mission, to do everything that it needs to do, including transmit information back to Earth and operate the instruments and so forth, gets on the order of a few hundred watts or so, something along those lines, which is not bad as these things go. And given the fuel that it takes to get something into orbit, to move it out to Saturn, to slow it down, and to ultimately maybe land something on the surface, you think about the size and scale and mass of a rocket, right, that takes off. Tens of thousands of kilograms of mass. What you actually land in terms of a science payload might be 50 to 100 to 150 kilograms or so, which means that hard choices have to be made. So the study that just happened and is basically proposed as a possible mission put a few different things on. It put a microscope, because I think any biological voices in the question, for them, that's the gold standard. We want to be able to actually see something that's alive. And so part of going through what I just described is to try to constrain, right? How much material will we have to look at? How much do we need to actually see? So a microscope is one thing. A mass spec is part of the package, because the counterpart to actual live things is the chemistry, the diagnostically biological chemistry that they've produced. Saying a mass spec could mean a whole range of things, and so there's other stuff tied up in what kind of front end separation do you do? What is the mass range? What's the mass resolution? All of these sorts of things. But generically, let's say a mass spec is on there. The last thing was an interesting choice. So there's a guy who has something that he calls the polyion theory of the gene. And if you think about what our genetic material is made of, it has basically a polyphosphate backbone. And what's interesting about the polyphosphate backbone is it can be bonded to sugars on both sides, and yet it still maintains a repeating negative charge along the backbone. And the argument this guy has made is that gives it very unique properties. And in particular, that repeating negative charge so much dominates the behavior of the DNA and RNA molecules in solution that it doesn't matter if you change the elements inside. So you can change G and C and A and T and do things that should affect the chemistry of the molecule, but really are only second order to this polyphosphate backbone. So the notion is, even if the coding elements are different, even if they're not our base pairs that we use, that in order to have genetic material, you may need something like this repeating charge polymer. Okay. And so the last instrument that was suggested is something called a MinION. So a MinION is basically, it's a field portable DNA sequencer, but its principle of operation is that it genetic material passes through these tiny little nanopores. And when it does, there's a charge difference that gets sensed. And the charge difference is, can be attributed to differences in A and T and G and C, but it's also responsive to things that may have different base pairs. And so the idea is that if you encountered one of these repeating charge polymers, you might know it. And I think that's kind of a clever approach, because it takes something that is, in our biochemistry very specific. So the choice of DNA and RNA is biochemically a very specific choice, but boils it down to a much more generic principle, and then finds an instrument that can actually make a measurement on that basis. So that's what is proposed to send, and actually I fairly well agree with that set of choices.
Okay. Does anyone else have questions? This has been very interesting talk. I have to admit I didn't understand everything, but the basic underlying idea is very fascinating. So on a daily basis, what is your work like? Are you lab in the field doing computations? And how did you, in your career, educational background, prepare for what you're doing now?
Yeah, good questions. So let me tackle the second one first. As John once said, I started in chemistry. That was my undergrad. I went into oceanography. That was my graduate degree. In the process of that graduate degree, I sort of picked up on the fly microbial ecology. So understanding how microorganisms interact with their environments, because there's such a dominant control on the chemistry of the system that you need to understand how they work in order to understand the chemistry. I came to NASA right at about the time that astrobiology started to become a thing at NASA. And it turns out that the way that the oceanography community approaches things, a very interdisciplinary, environmentally contextualized field, is the same kind of thinking that I think makes a difference in astrobiology. And so it was a good fit.
Oh.
And so I then sort of picked up planetary science on the fly, because it's hard to be at NASA and not get at least some of that.
Right.
When ocean worlds started to become a thing, all of a sudden, it's somebody who understands at least some of planetary science, oceanography, microbiology, and chemistry has a fairly useful skill set. It wasn't purposeful in any way at all, I'll guarantee you.
Right.
It's not like I set out to do that, but it
Yeah.
worked out pretty well. In terms of day-to-day basis, so I in the trajectory of my career, I was really surprised how quickly I ceased to be in the lab on a daily basis. And I really love it. Like I enjoy lab work. I feel like I have some utility with it. And yet it wasn't very long after my postdoc that really my work became finding the funding and doing the things that help support the people around me.
Yeah.
So much more of my day-to-day, as it probably is for you, Desmond, is I sit at my desk and write. I write papers, I write proposals, and the few occasions that I do get to go out to the field, I'm giddy. Like I love being in the field, because it's one thing to focus on, instead of the dozen balls that are usually in the air.
Right. Very good. So that's a good perspective for our students, because basically everything you learned along the way is positioned you to be where you are.
Mm-hmm.
Everything seems to just come together.
It's, I'm always amazed when I talk to people in my own department, whatever else, how many of them seem to have had some kind of similar wandering path.
Yes.
Like not terribly purposeful. Sometimes it's taking the opportunities that are available to you. Sometimes it's following your nose into what's interesting, which largely has been in my case.
Yeah.
And then some of it is finding yourself in the right place at the right time, and some of it is trying to figure out where your tools are most useful. What's the combination of what can I do and what I'm able to do and what I'm interested in doing. And if you can check all those boxes, you're in pretty good shape.
Yep, that's a good take-home lesson. All right, so thank you so very much.
Thank you all. This was
one
fun.
of the things we try to do is expose our students to chemistry wherever it is found. And some of these things are not taught in the textbooks and the formal classroom, but gives us a sense that chemistry is essential and central to a lot of, so.
It is the central science.
Yes, sir. Thank you so much.
All right. Thank you all very much. Bye-bye.
All right. Take care. Bye.